Day 6. Enzymes are life's biological catalysts. Welcome to day 6. Today we will explore the enzymes. They are biological molecules that make life possible through speeding up chemical reactions. What are enzymes? Enzymes are commonly protein in nature that act as a catalyst. They increase the speed of biochemical reaction without being consumed through this reaction. Each enzyme is highly specific. What does it mean? It means it only works on a particular molecules called substrate. How enzymes work? It contains an active site. This region of the enzymes where the substrate bind and form what is called enzyme substrate complex. Okay. Then reaction occur. Bonds are broken, bonds are formed. We remove hydrogen, we added hydrogen according to the reaction that is catalyzed by this enzyme. Product is released. Product is formed, then released. An enzyme is free to work again. Once the enzyme releases the product, it is free to bind a new substrate and do the chemical reaction and produce next product that will be released. This is often explained by two theories. The old one, it is called lock and key theory. It is an old one. It is based on, or it forms an explanation of the specificity of the enzyme, that this enzyme is work only on one specific substrate, and this explained due to the similarity, or for exact, it is the complementarity between the active site of this enzyme and the substrate 3 dimension. But there is a new observation that there is a there is a movement occur in the enzyme. There is a dynamic, not a fixed state, like state that it is we can imply from the lock and key theory. So another new theory called induced fit model. It explained that once the substrate is around the enzymes and come in contact, it improves its active site to bind better with this substrate. Simple example, when you wear a glove, the elastic glove is small for the size of your hand, but once you wear it, it takes the shape of your hand. This is simply what induced fit model present, which give a proper explanation for the dynamic motion that we observed in the enzyme and can't be explained only by the lock and key theory. Factors affecting the enzyme activity. Number 1, temperature. Too high temperature can denature these enzymes. They are protein in nature. What is meant by denaturation? It is the loss of the bonds that responsible for the secondary and the tertiary structure. So this enzyme lose its three-dimensional structure, which depend on its function. So high temperature denature the protein, lost its 3D dimension, lost its function as an enzyme. pH. Each enzyme has an optimum pH work through it. So most of our enzymes in our body and in our blood are optimized at 7.3, which is our pH in our body. Okay, but another enzymes that present in the stomach, its optimal pH is 2, while those work on the intestine, their optimal pH is alkaline. Okay. Number 3, substrate concentrations. More substrate is available, it lead to faster reaction. But until a one point, which is each molecule of enzyme is saturated with a molecule of substrate. So adding more substrate can't make the reactions go faster because saturation of the enzymes come through this concentration of substrate. Inhibitors. What are inhibitors? They are molecules that reduce the enzyme activity. Let's have an example about enzymes. Amylase, it breaks down starch in the mouth. Pepsin, it digests protein in the stomach. DNA polymerase, it builds DNA during replication. Lactase, it digests lactose, sugar in milk inside our intestine. Clinical relevance. Enzyme deficiency causes inherited metabolic disorders like phenylketonuria. Phenylketonuria, it is deficiency of the enzyme called phenylalanine hydroxylase, which is the first enzyme that work on the phenylalanine. This stops the metabolism of phenylalanine and causes this metabolic disorder. The enzyme deficiency caused by a genetic problem that is inherited from the parent to their offspring. Okay. Lactose intolerance. It results from the low level of the lactase enzyme that happened in some people. Enzyme inhibitors are used like drugs. AC inhibitor for treating hypertension. AC inhibitor, it stands for we having a system called angiotensin converting enzyme. This enzyme present in the lung, it converts the angiotensin 1 into angiotensin 2, which causes vasoconstriction in the blood vessel and stimulate the release of aldosterone, which lead to retention of sodium and accordingly water. So these enzymes elevate the blood pressures. So if we give an inhibitor for these enzymes, we can treat patients with hypertension. In summary, enzymes are nature catalysts. They represent the gift of the God for our body. They control the speed and direction of the life's chemical reaction with incredible precision. Tomorrow, wait for day 7, we will talk about vitamins and the coenzymes that formed from vitamins. These coenzymes are helper in biochemistry. Thank you.